join as we go along. I just want to make everyone aware that we are recording this session. So, and if you want a copy of the recording, I'd be more than happy to send that to you later on. Can everyone hear me? Okay. That's a thumbs up, so I guess you can. Okay, very good. So welcome everyone. Thank you for joining the online demo for the Accuva Cellect Laser Capture Microdissection. My name is Miles Bellamy. I'm the Director of Laser Capture Microdissection here at LAXCO. I have a little agenda here that I want to just go over briefly. So I'm going to talk a little bit about the background of the instrument myself, why LCM is important for researchers, different applications that people are using laser microdissection, specifically this instrument for, what our technology is and how it differs from some of the other technologies that are out there on the market, and then we'll do a live demo after that. So first off, my name is Miles Bellamy. I've worked in the LCM industry for a long time, since about 2002. 18 years experience working with LCM specifically, and a few other years working in imaging and high content and things like that. 13 of those years have been with the Arcturus technology. So that's working with Arcturus when it was a startup and through various acquisitions, most recently with when it was over at Thermo Fisher, and of course here at LAXCO. I've also worked for a couple of the competitors. I worked 3 years at Molecular Machines and Industries, or MMI, and 2 years with Leica. So I know the industry very well and all the different products that are available on the market. So to kind of give you a little bit of that background here is that I worked with Arcturus for about 6 years when it was a startup, through a couple of acquisitions, worked with MMI, then went to Leica, then did some high content stuff with PerkinElmer. And then in 2015, I was recruited by Thermo to come back and revitalize the Arcturus product line and generate a new product for that's going to be more competitive in the market, and that eventually became the Accuva Cellect. And I'll go a little bit more into the history in a later slide here. But when I was at Thermo, my main focus there was to take the older product and to make it into a new, more competitive and better product here at LAXCO. So LAXCO has been manufacturing the Arcturus Cellect since 2017. That's when we at Thermo contacted LAXCO to be our contract manufacturer and to develop and co-engineer the instruments. And basically what we did is we took what we had from the Arcturus XT and made that into took all the feedback from customers of what they'd like and incorporated that into the new instrument. We recently took over the sales of this product, and when we took over the sales, that's when I joined LAXCO and started working, supporting the instrumentation here as opposed to over at Thermo. And due to some trademark issues, we had to change the name to Accuva Cellect as opposed to Arcturus Cellect, but rest assured it's the same technology with all the same features. So a little bit of history about the Arcturus product line. So this was co-developed the technology was invented at the NIH, and then a partnership between the NIH and Arcturus commercialized that product for laser microdissection. This was a very, the first instrument was the Pixel, the Pixel 2, and the Pixel 2e. This was a very popular product line. There's about 600 or 700 of these out in the field. And this was kind of the industry standard for many years for being for laser microdissection. This allowed a lot of people to be able to enrich for certain cell types for their typically gene expression at this point, doing microarrays and qPCR and things like that. In 2003, people had started to ask for automation, so Arcturus launched the AutoPix. This was an automated version of the Pixel that allowed people to go in and work with a computer interface rather than doing a more manual laser microdissection process. In 2004, we incorporated a UV laser into the new product, the Veritas. This allowed a wider range of applications. So working with dense tissues and like bone and plant and things like that that weren't possible using just the IR laser. And I'll talk about the two lasers that we have in the system in a moment here. In 2007 was when the Arcturus XT was launched. This was based on a Nikon scope with the lasers and the fluorescence and all that pulled onto it. This was a very popular instrument as well. As you can see, it was commercial for about 10 years. And there's a lot of publications that go along with this instrument. It was very popular and very good one. However, you see there was a big gap here. About 10 years is pretty long for an instrument to be commercial. And so it started to show its age by some of the technology that was based in that. So as I mentioned, in 2015, I was recruited by Thermo Fisher to join and revitalize the instrument and make it a better product that's going to be more competitive in the market and better, more useful for researchers that are doing proteomic and genomic research. So we developed it in a cooperation with Laxco. As I mentioned, we reached out to Laxco as a contract manufacturer. Laxco has a lot of experience working in microscopy and being able to develop these kind of projects. So we contacted them, worked with them, and made the product that we have today. It was 2021 is when the first Arcturus Celects were sold. And then in 2023 is when we took over the commercial responsibilities from Thermo here at Laxco. And this change here was basically there was a leadership change over at Thermo. The Arcturus was a research use only product in a clinical division. So it wasn't a great fit in their organization. And when they had a leadership change, they wanted to find a better avenue for being able to sell this product. And so they reached out to us to be able to do that. And we still have a great partnership with Thermo for this. And I just want to mention that we have over 700 publications using this technology from a wide variety of different applications. I'll go into the applications in a little bit, but in oncology and neuroscience, there's a ton of publications and a wide variety of other applications as well. So why do we want to do laser capture microdissection? So the idea here is that if you have a tissue biopsy or some sort of sample where you need to enrich for a certain cell type, so maybe say you have a 10% tumor load on a biopsy and you want to go in and enrich for the cancer cells and leave the healthy tissue behind so it doesn't bias your downstream results. So laser capture really gives you the ability to go in and create a pure cell population from a heterogeneous mixed population. And why is that important? So if you were just working with a tissue biopsy, you're going to have a bias of healthy tissue that's going to influence your results, and you're going to have worse data. So this is just kind of an example of this. So on the left here, we have a whole tissue biopsy that was done via tissue scrape. And on the right here, we have a carcinomas that were done via LCM. So the carcinoma cells only. So this is the same sample, but if you have the whole biopsy, you see that you have a much different genetic profile. These are just 3 genes, but the whole genome sequence is going to be a lot different. As opposed to if you're going in and you're just looking at the carcinoma cells, you have a much different genetic profile. So if this is just kind of an example, if you're looking for the carcinoma cells and you have all this healthy tissue here, this is going to be a huge bias on your sample and your downstream genetic analysis. So this is the instrument. This is the one that we'll be looking at in just a moment here. So some of the applications that people use this for. So this was kind of developed for doing oncology research, but there's a lot of different applications that this can be used for. So oncology research, all kinds of different breast cancer, brain, and kidney and pancreas and all those kind of different cancers. Those can all be used with the Accuva to enrich for certain cell types. Neuroscience is another big area. So ALS, Alzheimer's, Parkinson's. A lot of people are doing that kind of research with our instruments as well. But some of the other ones that are people use it for are plant biology. So people can go in and capture different areas of the plants and do genomic research on that. Live cell microdissection, so being able to go in and grab differentiating stem cells and be able to reculture those. There's also other developmental biology applications we can do as well. Single cell capture, so people are now being able to do genomic and proteomic research on single cells and very small number of cells. This is primarily enabled by recent developments that have happened in technology in the downstream analysis. So next gen sequencing input requirements have come down, and so have mass spec requirements. So now there's kind of a resurgence in LCM that in these kind of applications, because people are able to use this kind of LCM samples in these kind of research now. And then another one is human identification. So being able to go in, say in a rape smear, and be able to grab the sperm cells and leave the epithelial cells behind, so that you can identify the perpetrator. So our system, there's a few systems out on the market. Our system, how it's kind of differentiated is that this is a purpose-built laser microdissection system. So this is built and designed specifically for doing laser microdissection. It's designed to be very compact, because I know that some of the bench space in laboratories is very valuable sometimes. Some of the other instruments, including our old instrument, the Arcturus XT, it was a microscope with a laser slapped on and a computer next to it and a fluorescence box next to it and all those kind of things. So what we did is we tried to make the footprint as small as possible by incorporating the computer, the LED illumination, and the lasers are all built into the same instrument. So that's a very small footprint. It's about 60 centimeters square on your benchtop. And what the unique feature about our system is that we have a dual laser design. So we have both an IR laser, a very low energy infrared laser that's used to capture cells, and then we have a high energy UV laser that's able to cut through dense material. So I wanted to talk a little bit about the two different lasers that we have in here, and when you would want to use one versus the other. So this is an experiment we call the sunburn experiment, and this is kind of showing when the lasers are best used. So this here on the left, this is capturing a large area using only the infrared laser. So this is using the infrared laser and going through and capturing all these cells. And over here we have the same area but captured with a UV laser. And what I wanted to show here is, so this is a bioanalyzer profile showing the 18S and 20S peaks for RNA quality. And you'll see that the RNA quality is very similar for both of these samples. You have very good RNA quality in your downstream genomic analysis, gene expression and things like that. That's going to be very good quality to go into those kind of experiments. So in this kind of situation, you could use either of the two lasers and be able to get good results. In this situation, you probably would use the UV laser on the right here, because this is going to be much faster than if you were using the UV laser. I'm sorry, if you're using the IR laser. But now if we get down to smaller cells, so this right here, this is a 30 micron diameter area as opposed to 650, so much smaller area. So these are individual spots all put together on the same cap. And you'll see that with the IR laser, you're still getting great RNA quality here, good solid peaks, no degradation. So the RNA quality is very comparable to what we saw with the large area. However, if you start to use the UV laser for these small areas, you'll start to notice that there's quite a bit of degradation here. And what this is this is a result of the high energy UV laser. So when the UV laser goes around and it cuts around a cell, it's creating it's a high energy laser. So there's going to be a little bit of leakage on both sides of the cut path. And this leakage is going to be damaging the biomolecules and the genomic, the RNA and the DNA and the proteins as it goes through. So the difference is that when you go with a large area, you have so much material that leakage is a very small percentage of the overall amount of material that's going into your sample. So it comes out in the noise. You don't even really see it when you're doing your quality control. However, when you get down to these small areas, the amount of leakage that you have on each side of the cut path is quite a bit more, and it's more noticeable. And so this is when you start to see the degradation, is because it's a higher percentage of the overall amount of material that you have in there is being degraded. So it starts to show up on your quality control. So in this kind of situation, you would really want to use an IR laser only, so that you still get good quality material out of your small samples. And another advantage of using the IR laser only is that when you're trying to grab adjacent cells, that you're not cutting through the cells that you want. So in this situation, we're trying to grab these small dorsal root ganglion, and these are so you can go in and you can grab these and then put these onto one cap, and then take that cap away, bring in another cap, and capture the large dorsal root ganglion, and do comparative studies out of the same sample. If you were using a UV laser to capture this cell, you would have cut through these cells, or vice versa. If you're capturing these cells, you would have cut through this cell. So you wouldn't be able to do this kind of comparative study if you're using the UV laser. So I want to talk a little bit about our technology and how the technology works. So the way that our technology works is we work with these caps. These caps are about 1 centimeter, and they sit directly onto the tissue on the biopsy. And what we do is we fire an IR laser through the top of this cap, and there's a polymer on the bottom that has a dye in it, and that dye absorbs infrared laser energy. And when it does that, it distends down and becomes adhesive. So it's going to stick to the cells that are directly underneath it and capture those, and then you can lift it up, and the cells will be attached to the face of the cap. We have a variety of different caps. There are 3 different types of caps depending on what the research needs are. So we have macro caps that are used for large areas. We have micro caps for working with different size tubes, so 0.2 mil tubes and 0.5 mil tubes. And then we have HS, or high sensitivity caps, that are used to capture very small areas and get very small spot size down to about 7 to 10 microns. And then we're also able to use a variety of different slide formats. So most of the industry is using a glass membrane slide, and what this is a pen membrane polyethyl naphthalate membrane, and it's only attached on the perimeter, and in the center it is free floating. What this allows you to do is go in and cut through the membrane and the tissue and pick that entire area up all at once, and this allows you to do very quick microdissection. However, we are able to also use standard glass slides. So this enables you to just put the tissue directly on the slide, and then capture the cells directly off of that. This can be good if you're doing something like a cell smear. If you have archive slides that are on standard glass slides, we can work with those. And also these membrane slides have a bit of cost. They're somewhere around $7 each. So standard glass slides are almost free. So if you're trying to reduce your per sample cost, you can use standard glass slides instead of using the membrane slides. And then frame membrane slides are another format that allows for more advanced applications. So in situations where you can't stain your samples, you need to work with wet samples, things like that. So we have the flexibility to work with all of these different slide formats. So again, just to talk about our technology a little bit. So again, we work with these caps. So you're going to put the cap directly onto the tissue biopsy. So to give you an idea here, here's the cap, this is the tissue, and this is the slide directly underneath it. So what you do is you bring the cap down, you fire the IR laser through the cap. There's a polymer on the bottom here, as I mentioned, that distends down and becomes adhesive and sticks to the cells of interest. And then you lift the cap up and the cell will come with it and it's stuck to the face of the cap. You then take this cap, bring it into a microcentrifuge tube that has a little bit of lysis buffer in it, flip it over, and then you can start your extraction directly off the face of the cap. And this is a scanning electron microscope image of single cell capture on the face of the cap. So again, this is the polymer. It's distended down and it's became adhesive and stuck to this cell, which is then lifted off the biopsy. So this is just kind of our workflow, is to go through and find the areas of interest on the computer screen. We have different formats that we can draw on the screen, and I will show all of this live in just a moment. Doing the cutting and capturing, and then being able to visualize the cells on the face of the cap. So in a moment, I'm going to show a video of this, a very brief video, and just kind of walk you through kind of the technology and how it works here. So again, the tissue biopsy, bring the cap down, put it directly onto the biopsy, and then using the computer interface, we're going to find the areas that we're interested in and draw around it. So this red line that we have here, this is a UV cut line. And then we're going to put some IR spots on here. And the reason for these IR spots is so that you can stick the tissue to the face of the cap. Because if we just did the UV cut, the tissue would just stay on the slide. So this allows you to stick to the cap. And so it placed the IR spots first and then did a UV cut. And so this is just showing the same thing again, just drawing around the region of interest. Place some IR spots to stick it to the cap.
Miles, can I ask a question?
Yeah, absolutely.
Thanks so far for the great overview. So how would that work for the glass slides? So I guess you would have to really rasterize over the entire ROI, or because I mean, it's much more challenging, right? If you don't have the membrane.
Correct. So if you were doing a glass slide only, you would have to fire IR spots on every single cell that you were interested in. And so that's kind of the when I showed the sunburn experiment earlier on, is why you would want to use the UV laser for large areas, because going through and rastering with 100 cell or 100 IR spots is going to take longer than just doing a quick UV cut. But if your sample is on a glass slide, then we can absolutely do that. I mean, we're talking, maybe a minute or something like that we're saving. So it's not a onerous way to do microdissection by doing LCM only. It's just this allows the ability to do it faster.
Thanks.
Okay. So we put the IR spots here, and then you can go through and you can place individual IR spots on the cells of interest. You don't have to use the UV in this situation here. So once you've done all your microdissection, you lift the cap up. You can visualize all the cells on the face of the cap, and then again, just take this into a microcentrifuge tube and do your extractions. Okay, and so now I think we're ready to go and do the live demo. Let me stop presenting here. Turn off that camera, and then we'll work over here. Okay, so you can all see the screen there with the software. I'm gonna very quickly. I just want to show you. So these are the LCM caps that we have, and then I have some slides here and some caps that I'm going to load in. So the first thing when you come to the instrument is that we want to load our samples. So just to kind of orient you what's going on here is that this is the stage where we're going to be working with. It has room for 3 different slides and 1 cartridge of caps, and the cartridges carry 4 caps each. These can all be hot swapped. So if you want to add more tissue to a single cap from different slides, you can stop and pull out one slide, put another slide in, and put the same cap down. The opposite is also true, is that if you have one slide that you want to do a comparative study of, say, carcinoma and healthy tissue, you can use one slide and put multiple caps onto that. So it's very flexible in how you want to do your research. So first thing I'm going to do is I present the stage, and I'm going to put the cartridge of caps in, and then put my slides in. So now I've got this all in here. I'll bring my so I'll tell it what I have in here. So I have 3 slides. I'll go to my first slide here. And so the first thing I want to do is get an overview of what this this tissue looks like. So on here we have what we call a overview image, and there's nothing there at this point. So first thing I'm going to do is acquire an overview image. So what it's going to do is going to go through and take a series of 32 images of the slide and tile them all together with XY coordinates, so that I can work with the overview image and be able to quickly go through and navigate through the slide. And so now I have an image up here of the whole slide, so I can quickly navigate around and find regions that I'm interested in. And so we have microscope controls here. So these systems are all fully loaded with full complement of objectives. So 2, 4, 10, 20, 40, and 60x. So depending on what I'm interested in, I can use any magnification. So let me find something in this region here. So I'll come in and 4x. And one thing I wanted to point out is you'll see this little red box here that's on the overview image. This is showing what the field of view is on the roadmap. So if I move around, you see that red box moves around with me. And then as I move to higher magnification, that red box is going to become smaller. Okay, let's say that something in this region is interesting. So I'm going to try to interrogate this area here. So the first thing I typically will do is I'll bring in a cap. As I mentioned, I have 4 caps in a cartridge there. I have 3 different cap types that I can use. So if you are working with macro caps, you would click it here, micro or HS, and that's going to affect the usable area. And so let me bring in a cap real quick, and I'll show you that. So as I press to bring a cap in, you'll see that the instrument is going to go over. It's going to grab a cap out of the cartridge, lift it up, and put it onto the biopsy. The center of the cap is going to be directly in the center of the field of view here. So you'll notice that on the overview image, there's now a green circle. And what that green circle is that's showing me where the edge of the cap is. So you'll see as I come to the edge of that, you can actually see the edge of the cap here. And if you were working with macro caps, you see that the circle gets a little bit bigger. With HS caps, it gets a little bit smaller. And so it knows what type of caps that you're using and shows you the usable area of it. So let's say that I'm interested in an area right here. So first thing I'm just going to quickly test the laser, make sure everything's working. Yeah, everything's working here. And so I'm going to go through and let's see, we want to do a UV cut. So there's various ways that you can interact with the software. So so far I've been working with a mouse. However, you'll see that we also have a pen. This pen is, so all the systems come with a large touchscreen monitor, high definition touchscreen monitor. The one that I have here is a 16-inch. The instrument itself actually comes with a much larger one, a 27-inch. This one travels around a little bit, so it's a little bit easier to work with a smaller monitor for that. But this is actually an artist's monitor here. So you see, as I move my hand around, it doesn't actually move. But if I use the pen, this is it works and interacts with it. The reason for that is so that people can put their hand on the screen, and it's not going to be making any motion. So you have to use your finger to actually be able, or the pen to actually be able to make a drawing. So let me say that I'm interested in say this area right here. So I'm gonna come through and make a drawing, and you'll see that it's making a red line around, and then it'll automatically put IR spots in there. If it's a particularly large area, I can go through and I can put other spots in there. I can also do the IR laser only. So say that I wanted to pick up this area with the IR laser only. So I can come through like this. And then I can go through and I can do individual cells as well. So let's say that I wanted to do some of these cells here. So once I've made all my selections, I can go cut and capture. But I do want to talk a little bit about the drawing tools that we have here. So in addition to the drawing of lines that I did here, we have the ability to do rectangles and circles of predetermined sizes or freeform, individual spot sizes, a line of IR spots. One thing here is the eraser button. So if I wanted to, if I made a mistake here, I could go through and I can erase an area. And then so long as I connect both of the ends, it will know that that's the same section that we're trying to capture. And then the hand tool for moving, and then a measurement tool so that we can measure how large these spots are. So we're looking at about 36 micron spots here. So now that I've selected my regions of interest, I'll go through and I can tell it to cut and capture. So I have the ability to fire only the IR laser, only the UV laser, or fire both of them. Under most circumstances, you would fire both of them, and you can do one or the other beforehand. It really doesn't matter in most situations, but there are advanced applications where you one is better than the other, and we have the ability to modify it easily for that. Another thing is that we have this is a tunable laser, so we're able to control both the IR laser power and duration and the UV laser power and duration. So we can go through and we can crank up the laser power if we're working with some dense tissues, like say bone or bone marrow, plant tissue, something like that, and turn down the UV laser to very fine if we're trying to do similar to the experiment I did with 30 micron spots. And if we're trying to use that with the UV laser, we could go through and make the UV laser very, very low energy to try to minimize the sunburn effect on that, the leakage. So let's go through and cut and capture this real quick. So one thing I wanted to mention is that the lasers are off by default at all times, and they only are turned on when they're being fired. So this helps to preserve the life of the lasers, because they're only being used for seconds to minutes in any particular experiment, and the rest of the time they're all they're off. So right now it's firing up the UV laser, and it'll go through and it'll make the UV cut that I just selected there. Okay, so we've done our cut and capture. So one of the advanced features that we put on this compared to like the Arcturus XT or old systems is this QC check button. And what this allows us to do is it's just going to lift up the cap and move it out of the way so we can see whether or not it's actually picked up the cells that we were interested in. So I'm going to do that now. So it's bringing arm in, it's picking up the cap. And then we can see that the area that we were interested in, it's all clean and none of the tissue. And of course, you can photo document as you go through. So I can take a picture real quick before and after, and then of the cap. Your images can have the annotations on there, or the annotations can be removed depending on which way that you want to do that. So let's go take this in and do a quality control check on this. So on the stage here, we have 4 quality QC stations where each of the individual caps can be visualized. So I'm going to take this and bring it to a QC station. Bring that into focus. And you can see all the tissue that we selected there. You can take a picture of that. And then at this point, we would present the stage. And then I would just take the cap off. I have a microcentrifuge tube that would have lysis buffer in here. I would put the cap into the tube and flip it over, bring the lysis buffer down, and then it's starting the extraction. And if you're working with protein or RNA, at this point, your RNA and protein is going to be safe, and you can continue doing other experiments, or just take it directly and do your extraction protocol. So that's the instrument in and its functionality in very brief format. So one thing, if you're working with proteins in RNA, is that you want to be working very quickly. So on most situations, you have about 15 minutes from when you've dehydrated your sample and put it onto the instrument to when you want to have it into your lysis buffer, so that the inherent RNases, proteases don't become active. So this is made to be able to work very quickly so that you can stay well within that 15-minute window. And of course, that window changes depending on the tissue type that you're working with. So at this point, I wanted to pause and see if I have any questions, and if there's any other things that you'd like to see the instrument do.
Yes, one question. So you mentioned the automation in the beginning. So what do you mean by that? So is there a way that you could, in a more streamlined fashion, really upload potential ROIs or single cell contours and then start the laser microdissection?
Yeah, so as part of the product pipeline, we have 3 features that we are working on. One of them is exactly what you're talking about. So just to give you a little bit of an idea, is that these are mainly from customer requests. Is one that people would like to be able to visualize these, have some sort of automatic selection of certain cell types. So there is a rudimentary one that's on there now. It's basically just colorimetric. But we're working on doing some things that are more in line with what you're talking about, is being able to look at what a certain cell type looks like, especially say we're working with neurons or something like that that are long and thin, as opposed to a breast cancer cell or something like that. So it's going to be more round. So being able to teach it, so these are the types of cells that I'm looking for, go through, find all these types of cells and select those, present them to me, and then I can say yes or no on each of those individual cells and be able to capture all those. So Yeah, so that is part of the product pipeline right now. As of right now, the cell selection would be a more manual process, though. But there's people are asking for this, and we're a small company, so we can make these kind of changes pretty quickly. Other ones that we're working on is being able to take an image from another source, of being able to annotate basically on your computer at your desk on a stain slide, maybe from a slide scanner or something like that. You have a serial section, and be able to bring that in and do all your annotations and bring that in and put that onto the unstained slide, so that you can, and then manipulate it around so everything lines up, so that you can do your microdissection on your unstained slide. This is especially if you're doing immunofluorescence or something like that. That it's very long and RNA damaging process. Being able to do that on a serial section is very valuable. We can do that part right now, but both would have to be on the instrument itself. What we're trying to do is make it so that you can do it at your desk and then do it. So it's just to make it a little bit more comfortable for customers.
And maybe a follow-up question. So the way the instrument is designed, does not allow to collect in plates in the foreseeable future, right? So you're kind of bound to the 0.2 ml or 0.5 ml tubes. Is that correct?
Yeah. So there is so we do have a protocol. So I do have microtiter plate here. I'm not sure if you can see it in my tiny little thing, but they do work. So these caps do fit snugly into a microtiter 96 well plates. So if you filled it up with fluid and put the caps on there, you can flip it over and do it that way. But I think what you're talking about is maybe like the Leica system, where you can drop it directly into the microtiter plates. Wells.
Exactly. That's what we're working with most.
Yeah. So that workflow is not currently on this system. We are developing a new stage insert so that we can do that protocol. However, at this point, that's not functionality on this particular instrument right now, but we are looking to do it that way. But the interface would still be basically to working with the caps. All right. Any other questions? Anything else that you'd like to see with the instrument? Okay. Well, I mean, we're pretty close to time, so if there's nothing else that people would like to see, I think we can end here. I'll send a thank you email to everyone and follow up.
Thanks a lot, Miles.
All right.
Thanks for the interaction. Thank you, everyone.
Hey, Miles,
Yeah.
just a quick question here. You said you could have a link for this recording. Would you be able to send that across, please?
Yeah. I'll get it from this is a Google Meet, so I'll get it from Google in typically about an hour, and then at that time I can make it available to everyone. Yes.
Great, thank you, and thanks for an excellent demonstration. Appreciate it.
All right. Thank you, everyone.
Thank you, Miles.
Okay. Thank you. Bye.
Thank you.